Welcome back to Chapter 5. In this second web lecture, we're going to be talking about proteins and nucleic acids. So just to remind you, you saw this slide last time. We talked about these macromolecules that are, for the most part, composed of long polymer chains of these building blocks called monomers. We're going to come back and talk about these last two macromolecules that do form polymers, the proteins and nucleic acids. So let's start with proteins. So the first thing to know about proteins is that they are incredibly diverse, both in structure and function. So proteins do the vast majority of the functions that cells carry out. So proteins account for more than 50% of the dry mass of most cells. A very important category of proteins are proteins called enzymes that work as catalysts to speed up chemical reactions. But proteins have many other important jobs in the cell, including things such as defense, storage, molecular transport, cellular communication, movement, and structural support. We'll look at each of these individually just very briefly. And cells produce tens of thousands of distinct proteins, so they are hugely diverse and carry out a huge number of functions. So they're a vital, tremendously versatile component of cells. The monomer units that make up these protein polymers are called amino acids, and amino acids have been found in meteorites, so that they can be produced under space-like conditions and were most likely very abundant during this chemical evolution phase before the beginnings of life on Earth. So here are some examples of the many and diverse functions of proteins. I mentioned their functions as enzymes to catalyze and speed up reactions. In many cases, they're going to bind to the two reactants, keep them close together to form products, and speeding up the reaction in that way. Defensive proteins such as antibodies that are going to help fight off intruders in your body and protect against disease. Storage proteins that maintain a source of amino acids for producing more proteins can be found in things like ovalbumin in eggs and milk proteins in mammalian milk. When we get to cell membrane structure and function, we're going to see that transport proteins are very important in regulating how materials can move in and out of cells and which materials are going to move in and out of cells. In addition to transport proteins and cell membranes, proteins are also responsible for transporting bulk materials large distances throughout a multicellular body. So for example, hemoglobin is going to be transporting oxygen from the lungs to other parts of the body. Proteins can act as hormones to coordinate responses in the body to the environment. So for example, insulin is a protein that's going to help regulate blood sugar levels due to variations in dietary intake. Proteins also serve as receptors. So for example, in the nervous system, the nerve cells are going to secrete these signaling molecules, which are then going to bind to these receptor proteins on the surface of the receiving cell, and that will help to determine how that cell is going to respond to that signal. Movement is a hugely important function of proteins in not just animals, but many unicellular organisms also. So these motor proteins are responsible for the undulations of cilia and flagella. And of course, actin and myosin are proteins that produce muscle contractions in animals. They also have an important function in structural support. So an example is keratin, the protein of hair, and also collagen, which is this incredibly strong fiber found in skin and other places where structural support is needed, and that is another example of a protein. Just to give you an example of how some of these proteins work, I'm going to show you a short video, and what I want you to notice is keep an eye on these contractile proteins, and what's going to happen is that binding to these other proteins in this is a cytoskeletal element in the cell. We'll talk more about that, but these sort of structural fibers, these contractile proteins are going to bind, and when they bind, they're going to change their shape, and when they release, they're going to change their shape again. So what you'll see is that this portion of the contractile protein is going to release and change its shape, causing this leg to swing forward, whereas this one is going to bind, and that's going to cause a change in shape that's going to cause this to move. And the overall effect is going to be that this contractile protein more or less just walks along that cytoskeletal element. So all of this diversity in protein structure comes from a set of building blocks of amino acids that have just 20 different kinds. So these 20 amino acids can be used, put together in different ways to create all of this diversity in protein structure. So a little terminology here. Polypeptides are unbranched polymers built from these amino acids. So any chain of amino acids linked together is going to be a polypeptide, but not all polypeptides are proteins. So a protein is a biologically functional molecule, so it's going to have taken some kind of active shape that allows it to have a function in an organism, and it can consist of one or more polypeptide chains. We think about the structure of amino acids. Amino acids are composed of a central carbon atom that's going to be bonded to a hydrogen with one of its bonds. Another bond is going to connect to an amino functional group. Another one of the bonds from that central carbon is going to be to a carboxyl functional group. And then the final one is going to be a variable group. We usually call it an R group that is going to be particular to each of these 20 amino acids. So here's a generalized schematic of what this looks like. We've got our central carbon, sometimes known as the alpha carbon. This is the non-ionized form of an amino acid. So here we have the amino group, carboxyl group, a single hydrogen, and the side chain. So that was the non-ionized group. In water, remember that the amino group acts as a base and accepts a hydrogen ion, and carboxyl groups behave as an acid in water and are going to donate their proton to the solution. And so in solution, this is what our amino acid is going to look like. We're going to have 3 hydrogens attached to this nitrogen. It's going to have a positive charge, and we're going to have our carboxyl group with a negative charge missing that hydrogen ion. So these resulting charges helps these amino acids to be very polar, right? We've got a very distinct positive and negative pole, which is going to attract the polar water molecules and keep it dissolved in solution. And it's also going to make those amino acids more reactive, having these full charges on either side. So these different side groups can be separated into different types. I do not want you to worry about memorizing which amino acid is of which type. You don't need to commit that to memory. But know what these general categories are. So there's a group of amino acids that are hydrophobic, so they're going to have nonpolar side chains. Again, we're only talking about the side chains. Obviously, the rest of the amino acid is going to be polar, but they're going to have nonpolar side chains. So we see mostly carbons and hydrogens. When we do have an electronegative atom, such as sulfur here, there's just one, and there's a lot of carbon and hydrogen to make the side chain overall nonpolar. Then we have a group of hydrophilic polar side chains. Then we have a couple of acidic, negatively charged side chains and a group of basic, positively charged side chains. And so the reason I want to bring these groups to your attention is later on when we start to think about mutations in the DNA that cause a change in a single amino acid, the effect that's going to have on the protein function is going to depend on whether that amino acid is exchanged for one with similar chemical properties or one with different chemical properties. So a mutation that causes one of these hydrophobic non-polar side chain amino acids for another hydrophobic amino acid, it's not necessarily going to have a big effect on the function of that protein. But if you change one that's hydrophobic for example, one that's acidic, you're going to have a huge change in the function of that protein. More on this as we go along, but this is the idea that I want you to keep in mind, is that there are going to be groups of these amino acids that are going to share properties in common. So if given a structural formula for an amino acid, you can determine its type by asking 3 questions. Does the side chain have a negative charge? If so, it has lost a proton in solution, so it must be acidic. 2, does the side chain have a positive charge? If so, it has taken on an extra proton, so it has to be basic. If the side chain is uncharged, does it have an oxygen atom? An oxygen atom, as you know, is going to cause the side chain to be polar, and so that highly electronegative oxygen will form a polar covalent bond, and thus it's going to be an uncharged polar, so it's going to have a partial charge to it. If the answers to all 3 of these questions are no, then you can say that it's a nonpolar hydrophobic amino acid. So let's take a look at the peptide bond in particular. How did these amino acids join together to form these polypeptides? Amino acids polymerize when a bond forms between a carboxyl group of one amino acid and the amino group of another. And as usual, this is going to be a dehydration reaction. So the resulting CN bond is called a peptide bond. These peptide bonds are kind of special. So these peptide bonds are unusually stable because something kind of unusual happens with the unpaired electrons of this nitrogen in the peptide bond. For reasons that are beyond the scope of this course, the unpaired electrons in this nitrogen become disassociated from the nitrogen and form sort of a pseudo-orbital that kind of is shared between the nitrogen and the oxygen and the carbon. So it behaves in many ways like a double bond. In addition to being stronger than a typical single bond, having this other shared pair of electrons, it also causes this nitrogen to form a planar shape. So this is going to not have the typical pyramidal shape that a nitrogen bonded to 3 other atoms would have. It's going to have a flat planar shape. If we look at this dehydration reaction, we can see that an oxygen is taken from the carboxyl group, joins with 2 of the hydrogens from the amino group. So that causes this nitrogen to be bonded to the carbon from the carboxyl group. And then we have now a carbonyl group on one end and the carbon bonded to the nitrogen, which is now bonded to just 1 other hydrogen plus water. So these peptide bonds form what we call a backbone. So this is going to be sort of the structure of the chain, and this chain is going to have these R groups, these side chains sticking out from it. So R group orientation such that side chains extend out and can interact with each other or with water. So here we have our backbone with all of these R groups attached to it in different ways. And these chains are going to have a distinct directionality. It's going to matter which way around the strand is. And by convention, we call the end with the amino group the N terminus. The end with the carboxyl group is going to be the C terminus. And by convention, we put the N terminus on the left so we can see the N terminus is sort of the beginning. The C terminus is the end. And when we are lining up our amino acids in order and looking at the sequence, we start with the N-terminus and then the amino acid associated with the side group on that part of the backbone, and then go through the amino acids through to the C-terminus with the carboxyl group. So another feature of these peptide bonds is that although these peptide bonds are incredibly stable and strong and form this planar shape, the chains as a whole will flex because the groups on either side of these peptide bonds are free to rotate. So that allows a lot of flexibility in the structure, and that's what allows the folding of these polypeptides into distinct shapes. So what do proteins look like? Remember, we've got this strong association in proteins between form and function, and proteins have just unparalleled diversity of size, shape, and chemical properties. So this allows them to serve a huge number of functions in cells because of that relationship between structure and function. So as a few examples, we can see collagen fibers here that are made up of 3 separate polypeptide strands that are sort of coiled around each other, giving it incredibly high tensile strength. We can see a shape here for a protein that interacts with DNA molecules that has a structure that perfectly coincides with the structure of the DNA that it's going to be binding with. Here we have sort of a donut-shaped protein that its function is to allow materials to seep through. So this is a porin that allows passage of materials through this hole. Porins can be very specific to particular molecules. We have aquaporins that admit water molecules through them but exclude other kinds of molecules depending on size and shape and polarity. Here we have a globular protein that has different regions that fit perfectly with other molecules that they're going to be interacting with, such as other proteins. So how do we get this diversity in shape? Where do these protein shapes come from? So I want you to know about these 4 levels of protein structure, and I'm going to emphasize a couple of aspects of this. So first of all, I want you to really understand what kinds of chemical bonds or intermolecular interactions are giving rise to each level of structure. In addition, I really want you to understand how each level of structure depends on the level of structure just before it. So secondary structure is going to depend on the primary structure. So the primary structure is its sequence of amino acids. The tertiary structure, the folding of the polypeptide chain is going to depend on the secondary structure. And then in turn, the quaternary structure is going to depend on the tertiary structure. So each of these ultimately depends on the primary structure, but they're going to depend on the level of structure just above it. So let's look at each of these levels of structure in turn. So again, the primary structure is the unique sequence of amino acids. So here we have our amino acid chain. So these are, remember, going to be joined together by peptide bonds. That's the bond that gives rise to this primary structure. The number of primary structures is practically limitless. You can have any number of amino acids in any combination of these 20 distinct types of amino acids. So the length can range from 2 amino acid residues to tens of thousands. So a huge amount of possible diversity between those 2 aspects. And then again, primary structure is fundamental to the higher levels of protein structure. So the primary structure is going to determine the sequence of these R groups, and the R groups are going to determine the properties and functions. So this order of different R groups and how they're going to interact with each other is going to be very important to the function of that protein. So as we'll see later in an example, a single amino acid change can radically alter the whole function of a protein, even a very large one. Secondary structure is formed by hydrogen bonds between the carbonyl group of one amino acid and the amino group of another amino acid somewhere else on that polypeptide backbone. So you can see this illustrated here. This will fold in different ways so that this carbonyl group is going to interact with this amino group, and this can come in a couple of different forms. So this is going to cause the polypeptide to bend, bringing these polar regions together to hydrogen bond. And this is going to happen in two ways. There are two basic types of secondary structure. One is an alpha helix, so this is going to coil around, bringing that amino group close to a carbonyl group distant in the chain, right? They're not going to be right next to each other, but this is going to bend in a way so that the carbonyl group from way down here in the chain, if you follow this up, is going to interact with the amino group way up here on that polypeptide chain. And these interactions are going to keep this coiled into an alpha helix. Another way that these interactions can occur is if this whole chain kind of does a hairpin turn and bends, and so the two strands are going to be interacting with each other. So again, these are going to be interactions between a carbonyl group and an amino group distant from each other, not right next to each other, but at some distance from each other on this carbon-nitrogen backbone. So you can see these interactions at various places along the lengths, and so they can actually be very far from each other in terms of the primary structure. So these are two different ways of diagramming the alpha helix and the beta pleated sheet. This is a typical ribbon diagram showing those two secondary structures. The tertiary structure arises due to interactions between these side chains, between the R groups, or between R groups and the peptide backbone. So something to do with the R groups that are attached to this backbone is going to give rise to the tertiary structure, and we usually think of this as the folding of the protein. So these contacts cause the backbone to bend and fold. Bending and folding contribute to the distinctive three-dimensional shape of the polypeptide in all of that diversity. I showed you some examples a little while ago. So there are 5 important types of R group interactions you need to know about. The first is, of course, hydrogen bonds. When you have polar regions on the side chains, you can get hydrogen bonds either between two different R groups or between an R group and one of the polar regions on the peptide backbone. The second is known as hydrophobic interactions, but keep in mind that it's not the hydrophobic regions that are actually interacting with each other. Basically, the hydrophilic areas in the water are kind of excluding them and forcing them together. So water is going to force these hydrophobic side chains together. Interaction that was mentioned in your book that I haven't really emphasized is one called van der Waals interactions. These are very, very weak attractions that have to do with the fact that electrons are always moving around in an atom. They're moving constantly, and so there are going to be these very brief, even in nonpolar molecules or nonpolar regions, nonpolar covalent bonds, there are going to be brief fleeting moments of positive and negative polarity just because of the location of the electron in that moment. So these, as you might guess, are extremely weak interactions, but when you have a lot of them working together at the same time, they can actually produce a measurable effect. Another R group interaction is a covalent disulfide bond, and this happens when there are two sulfhydryl groups on these side chains that are interacting with each other. And those two sulfurs are going to bond together to form a disulfide bond. It's going to have a big effect on the shape of that molecule, and that one is going to be fairly stable because that's a covalent bond. And then also when you have ions in your side chains, when you have full charges, now you can have ionic bonds between full opposing charges, and those are going to be relatively stable as well. So let's look at a diagram that shows some of these interactions. So here we have an example of a hydrogen bond between a side chain and the peptide backbone that's going to keep these two sections of the polypeptide chain close together. Here's an example of two hydrogen bond between two side chains, two of these R groups, between an amino group and a hydroxyl functional group that's going to again keep these two parts of the polypeptide chain close together. Here we see diagrammed a bunch of these hydrogen bonds maintaining the secondary structure of the coil, the alpha helix. When you have these nonpolar regions in the protein, this is going to tend to fold to the inside of the protein. Why? Because the polar regions are anxious to interact with the water molecules all around it, so they're going to kind of surround these nonpolar regions, and we call these hydrophobic interactions, even though they're kind of being forced together by the tendency of the polar regions and the polar molecules around it to bunch together. And then also here you get van der Waals interactions, those transient charges on nonpolar molecules that are going to tend to attract them together. Here you see an example of a disulfide bond. So these two are going to be covalently bonded together into this shape, and an example of an ionic bond. So these are going to tend to wrap and fold these proteins into their functional shapes. So some examples of final tertiary structure. Here is a tertiary structure composed mostly of alpha helices that are being held close together in different places by these kinds of interactions. Here's a tertiary structure composed mostly of beta pleated sheets, so you have more kind of flat strands folding up to form this shape. And then here we have one that's rich in disulfide bonds, and so this shape is going to be fairly stable. Finally, let's think about quaternary structure. So many proteins are composed of more than one polypeptide chain subunit. So we wouldn't actually call any of these polypeptide subunits a protein in and of themselves because they're not the unit that has a function in the organism. So we call this aggregate structure made up of several polypeptides. We call that the protein because that's what's going to be the functional form. So these polypeptide subunits are going to interact to form a single structure. An example is hemoglobin, which is made up of 4 polypeptide subunits. And these subunits are going to interact together using all of these same kinds of forces we've been talking about, these different intermolecular interactions, and that produces the quaternary structure. So the structure that's produced when these different polypeptide chains, each with their own tertiary structure, come together to form a larger functional unit. So some of this comes in the form of what we call molecular machines. This is going to become very important when we start to talk about cellular respiration. What you'll have is a series of different protein subunits that are all physically attached to each other, and each one of them is going to carry out a particular function in this sequence of chemical reactions. So that keeps these reactions happening all together in the same place and in the proper sequence. It's going to go from one part of this machine to the next part of the machine to the next part of the machine. So here's an example of how all of these higher levels of structure depend on primary structure. And so this is the example from your book of the sickle cell trait, which is caused by a single amino acid substitution in the beta globin gene. So going back to what I was telling you about the amino acids with different functional characteristics. So glutamic acid is one of those acidic side chains, highly polar, is being replaced with valine, which is one of the nonpolar amino acids. So you can expect this to have a big effect on the interactions, the folding, to create the final tertiary structure, because you've got an amino acid in there with very different properties. One is going to tend to hide on the inside of the protein. The other one is going to say, hey, water, here I am, come and interact with me. And so this is what ends up happening. You've got the normal shape for the beta hemoglobin subunit, and you've got kind of a weird shape, an unusual shape for the sickle cell beta subunit. When they come together in this quaternary structure with the alpha subunits, you're going to get the normal one that has sort of a smooth profile, and they're going to stay separate from each other, which enables them to effectively carry oxygen. This altered tertiary structure is going to cause the different proteins to aggregate together, and that's going to compromise its ability to carry oxygen. So you can see here micrographs of the differences in shape. This is the typical red blood cell shape that you normally see, and this is a sickled red blood cell. So how does this folding occur? What determines what kind of shape this is going to take? So the protein folding is often spontaneous. The hydrogen bonds and van der Waals interactions are going to happen because they're more energetically stable in that configuration. So often the folded molecule is just going to fold spontaneously because that is its energetically most stable state. But not always. Proteins can exist in different states of folding, and sometimes proteins called molecular chaperones are going to help out to help proteins fold correctly. Sometimes during the development of a protein, after it has been synthesized, it's going to go through various stages of folding before it reaches its final folded shape. Another thing that can happen is that proteins can exist in their disordered, not properly folded shape most of the time in a cell until they're actually needed for a particular action. And you'll hear explanations as we go through some of these processes that another molecule comes in and activates a protein. What that means is that it changes that protein from its improperly folded non-functional state to a properly folded functional state. And so that molecule is going to help somehow to get it folded in the right way so that it can be active. We'll talk a lot more about that as we get to some of these processes that involve activating proteins. So in addition to primary structure, physical and chemical conditions can affect the overall shape of these proteins. So alterations in pH, salt concentration, temperature, or other environmental factors can cause a protein to unravel and lose that functional folded shape. And this loss of protein's native structure is called denaturation. This is basically what happens when you cook food. You're denaturing those proteins. They're giving up their molecular structure, their tertiary structure, and becoming disordered and disorganized. So obviously an unfolded protein that's been denatured does not function in its normal way in the cell. In some cases, those proteins will fold back together as soon as those environmental conditions are removed, but in many cases that denaturation is permanent. So, when you cook food and put it in the refrigerator, it doesn't change back to being uncooked. So now let's think about nucleic acids. So nucleic acids' main function in organisms is to store, transmit, and express hereditary information. So this is the passing down of information through time from generation to generation. So what does this have to do with proteins? This primary structure of a protein, the sequence of amino acids in a polypeptide, is programmed. That information is stored by a unit of inheritance called a gene. So genes are made of DNA, which is a nucleic acid, and this is a polymer that's made up of monomers called nucleotides. So DNA is short, of course, for deoxyribonucleic acid, and that's going to be the main storage center for genetic information in modern cells. But historically, most researchers think that probably ribonucleic acid, or RNA, was probably the first life molecule and was the initial storer of genetic information in these earliest forms of life. So this is called the RNA world hypothesis. We'll talk more about this as we go along and think about the structure and function of RNA. And this hypothesis proposes that RNA was able to both store genetic information and also be able to catalyze its own replication. So stay tuned for that. It's an interesting story. So then once self-replicating molecules that can both store the information and catalyze its own replication came about, chemical evolution would have given way to biological evolution by natural selection. RNA molecules that were better at replicating themselves are going to become more common in the world. So the roles of these nucleic acids are familiar to most of you who have taken biology classes earlier. There are two types, deoxyribonucleic acid, or DNA, and ribonucleic acid. And DNA basically provides the directions for its own replication. We'll see how that works as we go along. And DNA also directs the synthesis of messenger RNA. This is what contains the information for the primary structure of polypeptides, the order of amino acids in a polypeptide. So DNA directs the synthesis of a messenger RNA. Messenger RNA controls protein synthesis, and this process is called gene expression. So illustrated here, we've got DNA synthesizing mRNA. The mRNA is going to leave the nucleus and join up with the ribosomes, which are sort of the workbenches for putting together these amino acids into polypeptides. So you may be familiar with this as being the central dogma of biology, DNA to RNA to protein. But what is a nucleic acid? A nucleic acid is a polymer of nucleotide monomers. And a nucleotide monomer is composed of three components. One is a phosphate group. The second is a 5-carbon sugar. And the third is a nitrogenous base. So both the phosphate group and the nitrogenous base are bonded to the sugar molecule. Ribonucleotides are the monomers of RNA, and the sugar, the 5-carbon sugar that forms this backbone, is the sugar ribose, which has a hydroxyl group bonded to the 2' carbon. Deoxyribonucleotides are the monomers of DNA. The sugar is deoxyribose, deoxy meaning without oxygen, and on that 2' carbon it has just a hydrogen instead of a hydroxyl group. So this is the without oxygen part. It's missing this oxygen atom. So both of these sugars have a hydroxyl group bonded to the 3' carbon. So let's take a look at how these are put together. So a little bit of terminology. A nucleoside is the term we use to refer to just the nitrogenous base plus the sugar. So this part of the nucleotide, just the carbon and the nitrogenous base. Again, in DNA the sugar is deoxyribose. In RNA the sugar is ribose. So here is our ribose with the hydroxyl group in RNA and just the hydrogen by itself on the 2' carbon in DNA. And we use the term nucleotide to refer to this whole unit, including the phosphate group, the 5-carbon sugar, and the nitrogenous base. There are two different kinds of nitrogenous bases. One group is called purines, which we have here on the bottom, and these are composed of 2 rings, including carbon and nitrogen. So remember, these corners that aren't labeled are always carbon. So it goes nitrogen, carbon, nitrogen, carbon, 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 nitrogen, nitrogen, nitrogen. And these double rings are the purines, and there are two different kinds, guanine and adenine. The pyrimidines have a single ring composed of 6 atoms of a combination of nitrogen and carbon. So again, nitrogen, carbon, nitrogen, carbon, carbon, carbon. And there are 3 different kinds. 2 of them occur in DNA, so cytosine and thymine occur in DNA, and in RNA, instead of thymine, you get uracil as well as the cytosine. When these hook up, this is what they look like. So here we have the sugar rings with their phosphate groups attached. These yellow circles indicate the phosphate groups and the sugar rings, and then these are the nitrogenous bases attached to the side of this backbone of phosphate and rings of sugar. So these nucleotides are going to polymerize again using a condensation or dehydration reaction. And in this case, this bond is called a phosphodiester linkage. So it's going to occur between the phosphate group on the 5' carbon of this sugar ring. So here's the 5' carbon. It's going to, the phosphate is attached to it, and that's what's going to react. And then the hydroxyl group on the 3' carbon of that sugar ring is going to react with the phosphate group on the 5' sugar of the next nucleotide. So here we take one hydrogen from the hydroxyl group, we take this whole hydroxyl group from the phosphate, and those are going to come off as water. The oxygen on this 3' carbon is going to be bonded to the phosphorus atom that's attached to the 5' carbon on the next ring. So here's a 5' carbon attached to the phosphorus, and this is where that link is going to occur. So here we see the oxygen bonded to both the 3' carbon before and the phosphorus atom, which is going to be attached to the 5' carbon on the next nucleotide, releasing water in the process. So again, this is going to come in two types. Ribonucleotides are going to polymerize to form RNA. Deoxyribonucleotides are going to polymerize to form DNA. So just as we said for these polypeptide strands, the DNA and RNA strands are going to have a particular direction due to the bonding between something attached to the 5' carbon on the sugar and something attached to the 3' carbon on the sugar. So 3' and 5' carbons are joined by a phosphodiester linkage, leading to an overall 5' end of this polynucleotide and a 3' end. This is going to be extremely important when we start to learn about DNA replication and gene expression, because all of these processes are going to happen in a particular direction along this DNA strand. So one end has an unlinked 5' phosphate group, the other has an unlinked 3' hydroxyl group, and this is someplace where we can add on more nucleotides if necessary. The order of nucleotides forms a primary structure, so the sequence is generally written in the 5' to 3' direction, and that reflects the order that nucleotides are added to a growing molecule, always from 5' to 3'. So what is the ultimate shape and structure of these DNA molecules? So DNA molecules, remember, they have a primary structure that's determined by the order, the sequence of the particular kind of nucleotide that is attached. But then they're also going to have this secondary structure that's going to consist of two polynucleotides spiraling around an imaginary axis, forming what's known as a double helix. Most of you are probably familiar with this shape, but imagine taking a rope ladder and just twisting it. The backbones, these two polynucleotides, are going to run in opposite directions. So one will have the 5' end on one end, and the other one will have its 5' end on the opposite end of the molecule. So we call this arrangement anti-parallel. The two strands are going to run anti-parallel to each other. One DNA molecule is going to include many genes. When we get into eukaryotic organisms with chromosomes, a chromosome is one single humongous long strand of DNA that's going to have lots and lots and lots of genes on it. And only certain bases in DNA are going to pair up and form hydrogen bonds. So it's going to be adenine, always with thymine, and guanine, always with cytosine. And this is called complementary base pairing. And for those of you who may be fans of 70s and 80s heavy metal, this is a good way to remember this, ATGC. So this complementary base pairing makes it possible to generate two identical copies of each DNA molecule in a cell preparing to divide. The two strands just separate and then polymerize another complementary strand, creating two identical DNA molecules. So why do they pair up in this particular way? So there's a limited amount of space in this helical structure between these two phosphate sugar backbones. And so if you get two purines with their two rings, there's not enough space for them to effectively hydrogen bond. If you have two pyrimidines with their single rings, they don't get close enough for those two hydrogen bonds to interact effectively. You get just the right distance from each other if you have a 2-ring and a 1-ring next to each other. And that's what's going to cause that complementary base pairing, always a purine with a pyrimidine. So if we look at the nature of these hydrogen bonds with guanine and cytosine, it's going to form 3 for each pairing between an amino group and a carbonyl group. In the first and third interaction, and then this one in the middle is going to be between the hydrogen attached to this nitrogen and one of the nitrogens in the ring. Between adenine and thymine, there are going to be just 2 hydrogen bonds, one between an amino group and a carbonyl group, and then one again between a hydrogen in a polar covalent bond with one of the nitrogens on the ring and the nitrogen on the other ring. So in double-stranded DNA, the backbones have to run in anti-parallel directions again. So here we have 5' to 3' on this strand, 5' down here, running to 3' in the opposite direction on the other strand to make this work and come together in this particular way. So because of all of this hydrogen bonding between these complementary base pairs, this makes DNA very, very stable. It's very resistant to chemical degradation. It makes it a very reliable source for genetic information, to store that genetic information and make sure that it gets copied with very high fidelity. So stable molecules such as DNA are not very good at catalyzing reactions. So DNA has never been observed to work like an enzyme to catalyze a reaction. So biologists think that probably the first life form was based on RNA rather than DNA. It's a less stable molecule, but it's a much more flexible molecule, that lower stability makes it more flexible and allows it to take on more functions. So RNA, like DNA, has a primary structure consisting of a sugar phosphate backbone formed by phosphodiester linkages and 4 types of nitrogenous bases extending from it. So a very similar primary structure. But because RNA contains ribose instead of deoxyribose, the hydroxyl group on the ribose is going to be much more reactive and much more polar than the hydrogen in deoxyribose. And that's going to make RNA much less stable than DNA. And of course, the other difference is that RNA contains uracil instead of thymine. So what this means is that RNA molecules can also have tertiary structure. They're not going to be locked into this. They tend to have a helical shape, but they're not going to be locked into this double helix with the base pairing and all the hydrogen bonding. So this forms when the secondary structure can fold into more complex shapes. And that's due to the higher reactivity of that hydroxyl group in the sugar. So as a consequence of this, RNA is much more diverse in size, shape, and reactivity than DNA, which makes RNA highly versatile. It works almost as well as DNA as an information-containing molecule. It does have the complementary base pairing, but not locked into that one molecule, but it still has that capacity of self-replication through complementary base pairing. But it also, because of its ability to fold up into tertiary structure, it's also capable of catalyzing reactions and working similar to a protein enzyme, and we call these ribozymes, RNA molecules that are capable of catalyzing reactions. So what does this mean? It means RNA is capable of self-replication. It's got the information stored in that sequence of nucleotides that can be used to make copies of itself due to complementary base pairing. But as we see here, it's got to go through double-stranded intermediates. So if an RNA is going to make a copy of itself, it needs to polymerize a complementary strand. So here we have C pairing with a G, U coming to pair with an A, and then something needs to catalyze this polymerization to form these nucleotides, these individual nucleotides into a strand. We know that RNA itself is able to do that through its ribosome action. So then we have the template strand and the copied strand that are going to separate from each other. Now, the copy serves as the new template that's going to attract the individual nucleotides. Needs to catalyze the polymerization of those nucleotides to form them into a polynucleotide. Then we have this intermediate double strand that has the new copied strand, which is going to be identical to that original template strand because of the complementary base pairing. And so now we have a new copy strand and a new template strand that can go on to synthesize more copies of itself. So these are the double-stranded intermediates that it needs to go through twice to get a copy of this original template strand. And we actually see this process currently in some RNA viruses that reproduce their RNA in this way. So according to the theory of chemical evolution, there was this intermediate stage that involved what we call a naked self-replicator. So this is a self-replicating molecule that's not enclosed in a cell membrane. To copy itself, the first living molecule had to, number one, provide a template that could be copied, and number two, polymerize monomers into a copy of that template. So it needed to have some sort of catalyst to create those phosphodiester bonds. And so we saw that RNA is capable of both of these processes. So most researchers propose that the first ever life form was actually a self-replicating strand of RNA. And three of the characteristics of life are accounted for under this scenario. Information processing, so replication of the information contained in that RNA molecule. Replication of hereditary information, so being able to copy that information that's stored. And then evolution by random changes in nucleic acids, so any kind of changes in the nucleic acid that make it better able to catalyze that polymerization of new strands is going to increase its ability to self-replicate. And so the idea with this RNA world hypothesis is that eventually, due to these random changes in the nucleic acids and different combinations of the primary structure in the RNA, in RNA world, we start with RNA, we create a complementary strand that is eventually going to code for this polypeptide primary structure, the sequence of amino acids, and also perhaps be able to catalyze the peptide bonds that form this protein. So there's been a lot of research in this area. The discovery of one of the first discoveries of these ribozymes was actually a ribozyme that was able to catalyze the polymerization of RNA, showing that there's at least the capacity within RNA to work as a catalyst for that particular reaction, which would have needed to be the first step in using RNA to produce more RNA. And then lots of exciting discoveries about how we can simulate different conditions in the lab, both based on the substrate metabolism model, also models involving an environment with undersea volcanoes that are emitting different compounds into the sea around it. And researchers have been able to produce RNA molecules that can catalyze their own replication up to not the entire RNA molecule, but definitely portions of that. So a lot of research is supporting the viability of this model. And then, of course, this eventually led to the chemical evolution of a DNA molecule that's going to be more stable and store its genetic information more reliably. That would then use that as a template for an intermediate RNA that's going to give the information for how to build that protein. So finally, just to wrap up both of these topics together, the protein and nucleic acid segment, I want to talk a little bit about genomics and proteomics and how this is an absolutely transformational field in biology and answers so many different kinds of questions within biology. So historically, once the structure of DNA and its relationship to amino acid sequence was understood, biologists were eager to try to decode these genes and figure out how genetic information ended up being translated into a protein sequence. So the first chemical techniques for DNA sequencing were developed in the 1970s and have come a very, very long way since then, all the way up to our high throughput methods that can actually analyze DNA much, much more quickly nowadays. So bioinformatics is the name for the field that uses computer software to process these huge amounts of information, these huge data sets that allow us to sequence many genomes. And analyzing large sets of genes, or even comparing whole genomes of different species, is called genomics. So remember that genome refers to all of the DNA that an individual possesses. There's a similar analysis of proteins called proteomics that examines the sequence of amino acids in proteins and how those differ in different species. And this has allowed us to answer a huge number of questions and opened up whole new fields within biology. So one example of how this genomic approach has helped answer questions in evolutionary biology, whales and dolphins, the order Cetacea, has always been a huge enigma because they're so highly modified morphologically that it was very hard to figure out what groups of land mammals they might be related to. And so these genomic methods have allowed us to identify that actually hippopotamuses are the closest living relatives to all of the whales and dolphins. And so this was a very, very difficult problem to unravel until we had these genomic methods. Looking at patterns of dispersal and migration amongst early humans, how different groups of humans are related to each other. We've been able to use small stretches of DNA recovered from the remains of ancient ancestors to answer a lot of those kinds of questions. Obviously, medical science has benefited hugely from these. We're able to identify lots of risk factors for different diseases by looking at the genomes of individual people. Conservation biology, identifying which groups of organisms constitute kind of a genetic population. What should be the unit of conservation? Do we want to mix this population with this similar population over here, or is there distinct genetic information in one of those populations that needs to be preserved? Do we have a crisis of genetic variability in one population? Would it be better to interbreed with another population to restore that genetic diversity? All of these are questions that we could not even begin to answer without this field of genomics and genetic analysis. Species interactions, and microbial biology in particular has benefited hugely from this, just in being able to identify different species of microbes. We look at the genetic differences between them, but also interactions between plants and different microscopic fungi and microbes in the soil are allowing us to understand something about which particular organisms these plants are interacting with, what kinds of benefits are they getting from it, and this can help us to improve crop plants and help us with conservation efforts as well. So hugely important field. There's a section on this in the book with more detail, but just to touch on this briefly. So DNA and proteins act as tape measures of evolution. Can answer this question, for example, that we had about the whales and who their closest relatives are. Sequences of genes and their protein products document the hereditary background of an organism. So we can look at these linear sequences of DNA molecules that are passed from parents to offspring. And we expect that more closely related individuals are going to share more of these DNA and amino acid sequences with each other than either of them will with someone who's not as closely related. We can think of that within the same species. You're going to have more similarity in your DNA and proteins with your sibling than you are with a second or third cousin, for example. But we can extend this concept of molecular genealogy to relationships between species. And this is exactly how we figured out this whale problem. Molecular biology has added a new measure to the toolkit of evolutionary biology. We're no longer constrained at looking at the bodies and the morphology of these species. We can look into their molecules. And so, for example, if we want to know whether a human is more closely related to a monkey or a gibbon, which is a lesser ape, we can analyze, in this case, this is a sequence of amino acids in the beta-globin gene, or the beta-globin subunit of the hemoglobin protein. And we can compare the amino acids in the sequence. We can line these up. We call this aligning the amino acid sequence, and see where we have variation in the amino acid sequence. So we can see here in this first position we have valine for all three of these. In the last position we have histidine, and we can look for places where there are differences. So for example here, humans and gibbons both have S, monkey has N, so these are more similar between humans and gibbons. Similarly in this position we have the same in human and gibbon, different in monkey. And if we go down amino acid, I'm only giving 3 examples. Again, similar in humans and gibbons, different in monkeys. If we gather a whole bunch of these differences together and analyze them, we can see the percent similarity between these pairs of species and make a determination about who's more closely related. Spoiler alert, humans are more closely related to gibbons than they are to monkeys. So finally, this is the same slide I showed you at the end of the last web lecture, making that connection between this list of functions of all living organisms. I realized at the end that I had stopped 1 click short of this list because I had added on this growth and development, which wasn't on my original list, but it's also really important. And so these structural carbohydrates, cellulose, chitin, things that maintain cellular structure are very important to growth and development. And also steroids in their role of stabilizing cell membranes and also directing development as hormones are going to be hugely important there. But going on to the material from this chapter, proteins are going to be found everywhere. They do almost every possible job in the cell, regulating the movement of molecules into and out of cells as part of these transport proteins, catalyzing reactions and metabolism. I'm sure I'm not even going to touch on all the different roles that proteins play, but important enzyme functions for this transformation and use of energy. Reproduction, obviously nucleic acids have a big role in that transmitting of information through space and time. Proteins also, in the form of enzymes, catalyze those reactions, but also signaling and communication. These many hormones and transmitters, as well as the receptors for those transmitters of information, are going to be proteins. Ability to sense the environment and respond, particularly in movement and muscular reactions to the environment, proteins are critical. The ability to evolve. So nucleic acids play a big role in generating the variability in populations that are going to allow them to evolve, mutations, changes in those nucleic acids, and thereby the protein sequences are going to be a big factor in this ability to change heritable traits over time and evolve. And then if we add growth and development back into this list, nucleic acids, remember, are not just going to store that information, but they direct development. They're going to direct what proteins are expressed at what time in the life cycle and where and how much. And so both the nucleic acids and the proteins are going to be super critical to that function of living things on Earth.